Hello everyone, welcome to our channel Biochemistry for Student. In a previous class or a previous video, we have gone through the recombinant DNA technology part 1. In that, we have learned about important biological tool that is required for the actually recombinant DNA technology process. In part 2, we will going through the recombinant DNA technology process and what is the actual practical application of this technology. So, in a previous video, we have discussed about restriction endonuclease, that is known as molecular seizure, and it act as a act on a particular site in the DNA that is known as restriction site, having palindromic sequence, and they produces the sticky end as well as produces the blunt end. Then we have gone through the complementary DNA, how it is synthesized, how we get this complementary DNA with the help of reverse transcriptase. Then we have discussed about the vectors. There are plasmids, bacteriophage and cosmid, and they have different properties, and based on that we use suitable vectors for the recombinant DNA technology. Now we will discuss the basic procedure for the recombinant DNA technology. Basic, if we talk about this technology in simple terms, then in this technology we multiply our gene of interest, so we get that particular gene or multiple copies of that gene, or we get the protein that is due to expression of that particular gene. For that, we first isolate a DNA or a gene from the human cell, that is also known as gene of interest. Then this gene of interest that will be inserted into a plasmid, that or whatever the vector we are going to use. Then this vector carrying our gene of insert will be introduced into the host cell. We will allow this host cell to multiply in a culture, and then at the end what we get is a multiple copies of that particular gene, or if the gene is going to expressed, then we will get the multiple copies of that particular protein. So this is the basic process of recombinant DNA technology. Now we are going to learn each and every steps in detail. So there are 6 basic steps that are common to the most recombinant DNA experiments or the techniques. First one is isolation and the purification of DNA. For that, both vectors and the target DNA molecules will require to continue or to start this process. The target DNA molecule can be prepared by varieties of method like synthetic DNA or by the synthesis of complementary DNA, or we get that DNA directly from the gene library, and accordingly we get vector isolated based on the suitable vectors isolated from the bacteria for the recombinant DNA technology. The second step is that cleavage of the DNA at particular sequence. Once we get that plasmid, now we have identified a particular gene with what we want to multiply, then we have to cut it at a particular sequence, the cleaning. So the cleaving DNA to generate fragments of defined particular length or with a specific endpoint is a crucial process of this recombinant DNA technology. The DNA fragment of interest, that is called insert DNA, or it calls a target DNA. So in the laboratory, the process, the cleaving of DNA, is usually done with the help of restriction endonuclease enzyme. We use the same restriction endonuclease enzyme for the vector as well as for the gene to get on the DNA of the human to get the particular gene of interest. Now And third step, that is ligation of DNA fragments. A recombinant DNA molecules is usually formed by cleaving the DNA of interest to insert DNA. So by cleaving the DNA of interest to insert DNA, that is called target DNA, and then ligating this insert DNA to vector. So there will be insertion of our this target gene to the vector DNA, and what we get is called recombinant DNA, or it is also known as a chimeric DNA. So the DNA fragments are typically joined using DNA ligase enzyme. So as I told you, what we get is a gene of interest from the human DNA. We have plasmid as our vector. Both the DNAs that will be acted upon by the same restriction endonuclease enzyme. You can see here it is EcoR1. It produces the sticky end here, both in the plasmid as well as in the human DNA. Now after that, there is the process of annealing. So this our gene of interest will get attached to this plasmid with the help of ligase enzyme, DNA ligase enzyme, and what we get is chimeric DNA or the recombinant DNA. Now the next step is what we have got this recombinant DNA, that is a vector with our target gene. We have to introduce this into the compatible host cell so we can multiply the copy of that recombinant DNA. In order to be propagated, this recombinant DNA molecule that must be introduced into a compatible host cells where it can replicate. So this recombinant DNA can multiply in the host cells. So the direct uptake of foreign DNA by host cell is called genetic transformation, and the recombinant DNA that can be packed into a virus particle and transferred to host cell by the process that is known as transfection, okay. So there are various methods available for insertion of this vector carrying our gene of interest into the host cells. That is, the most commonly is the hypertonic solution with the chloride. We put the host cells and these vectors into the hypertonic chloride solution, and that will allow the chloride channel to open in that host cell wall, and that will allow this vector to enter inside the cell. The other methods like polyethylene glycol method, electroporation, microinjection, this liposome mediated gene transfer, and so on. There are varieties of methods are available. Now after inserting this vector to the host cell, we will allow the replication and the expression of recombinant DNA in the host cells. The cloning of these vectors that allow insert DNA to be replicated and in some case expressed in a host cells. So whatever the target DNA we have inserted through vector, it will replicate in some cases. So we get copies of that particular DNA, insert DNA, or it may undergoes expression, that is called genetic expression, in that particular host cell, and will get protein. The ability to clone and express the DNA efficiently, The ability to clone and express the DNA efficiently depends on the choice of appropriate vector and the host. That is a crucial thing for this recombinant DNA technology. Now, the identification of host cells that contain recombinant DNA of interest. This is very important step. Now here what happened? We have developed our chimeric DNA or the recombinant DNA, that is a vector carrying our gene of interest. Now when we introduce this vector into the host cell, there are three possibilities. The first one, that the host cell will get our vector with a recombinant or with the gene of interest. Okay, that is what we require. Second possibility, when we introduce this gene of interest to the vector, not all the vectors will take that gene. There may be few vectors or the plasmid, particularly if we talk about vector, then the plasmid will not take up that our gene of interest, and the bacteria will take that plasmid without gene of interest. And the third possibility, the host cell will not take any of the plasmid, that is itself a normal plasmid or the plasmid having our gene of interest. So out of these three, we want these host cells to multiply, so we can get a multiple copies of our gene of interest. So this, when vectors, usually this can be done by certain parameters. So when vectors usually contains easily scored genetic markers or gene that allows the selection of host cells certain characteristics that have taken by the foreign DNA. We can identify this transformed host cells. Transformed host cells means the host cells having our gene of interest by antibiotic resistance. When they take this particular plasmid or that having our gene of interest or the insert DNA, they develop certain particular antibiotic resistance, or they lost certain antibiotic resistance in selective medium. Based on that, we can identify. This shows certain changes in visible characters, or there are certain biological activities may develop that can be assayed from that colony hybridization, or by the blotting test we can identify these are the host cell. They are transformed with the plasmid having our gene of interest. So here this is an example how we can identify the transformed host cells with our gene of interest by by losing the antibiotic resistance. Here you can say this is a plasmid. When it take our insert DNA, here you can see it, when it take our insert DNA, it loses a one type of resistance here. This plasmid is resistant to ampicillin and resistant to tetracycline. When it take our insert DNA that will be introduced into the part or the gene that is resistant to ampicillin, this resistance will go and this tetracycline resistance will continue. So from that colonies that grow on a medium having, as I told you, three types of host cells, one with having vectors with our desired gene, one with only vector but no gene of interest, and third is without vector. So we can identify the colonies by this antibiotic resistance. Those who will not have any plasmid will kill by the both the antibiotic, either ampicillin or tetracycline. Those having a plasmid without interest like insert DNA, there will be resistance to both ampicillin and tetracycline, and the host cells having our gene of interest, they will lose their resistance to ampicillin. Okay, so based on this insertional inactivation of antibiotic resistance, we can identify our transformed host cells. So this is the process. So the DNA cloning in a plasmid vector permits amplification of DNA fragments. As I told you, that we get messenger RNA, we go through the process and produces the complementary DNA. We insert that DNA into the plasmid, that is a vector that will be taken up by the host bacteria. We will allow the suitable host cells to multiply. We get multiple copies of either gene or the proteins. So this is all about the recombinant DNA technology process. Now we see the applications of this recombinant DNA technology. What are the uses of this recombinant DNA technology? There is a very long list of these applications, like analysis of gene structure and expression. There are production of certain pharmaceutical products like drug and vaccine. We get some genetically modified organisms and certain applications in medicine we will see. Now the first thing is, by using the specialized recombinant DNA technology, we get a vast amount of DNA sequences in the human genome, and the entire genomic sequence of the human being have so many experimental role. So it is very important. And this enormous data, what we have, a very big data of having these DNA sequences that been stored in a two primary data bank. That is, one is the Gen Bank, that is at the National Institute of Health, Bethesda, Maryland. The second is the EMBL sequence database at European Molecular Biology Laboratory at Germany. So from that we have a large human genome data, and we can get whatever the information regarding the human genome from this. The pharmaceutical application, this is a very important, we can say it's a blessing for certain diseases. We are having this technology. Some pharmaceutical applications of DNA technology, like large scale production of human hormones and the other proteins with therapeutic use. The most important one is insulin, that is commonly used in the patient with the type 1 diabetes mellitus, or in the later stage in type 2 diabetic mellitus. So it has a very important role, or it is also useful for the production of safer vaccines. A number of therapeutic gene productions is done in the pharmaceutical companies, like insulin, interleukins, interferons, growth hormones, erythropoietin, coagulation factors, and so more. A list of products we are getting by the recombinant DNA technology. The pharmaceutical companies are already producing molecules made by the recombinant DNA technology to treating the human disease, and this recombinant bacteria, they are used in the production of human growth hormone and in human insulin. This is, you can see the products for varieties of diseases produced by the recombinant DNA technology. Okay, so it is very important, and we can say a blessing for treating so many diseases. We can get safer vaccine with the help of recombinant DNA technology. In the previously, what we are using for the vaccination was a virus, killed virus or attenuated virus, having chances of infection. But with the help of recombinant DNA technology, we can use only epitome or the gene that is responsible for producing the immunity. We isolate that particular gene, we introduce that gene into the harmless virus, and again that will be inserted into the human being as a vaccine. So our body will produce only immunity against the viral protein that is responsible for the disease production, and actually the person will not get harm because of this harmless vaccinia viruses. So it is one of the safer way to produce the vaccine. The other important role is production of or the changing a revolutionary change in the agriculture. The plants with genetically desirable trait we get, for example like herbicide or the pesticide resistance corn and soybean, that decreases the use of chemical insecticides and increases the production. The golden rice, that is the beta carotene, so we can easily remove vitamin A deficiency that is responsible for the causing blindness. Here there is again a list of altered plant like rice with the beta carotene, canola with the high laureth oil, s- the delayed ripening tomatoes that extend the shelf life. Okay, elongated sweet pepper that gives improved flavor and easier to slice. The altered cotton fiber, the plasticized fabric. So you can see we can modify crops. We can modify crops for desirable effects. Then comes the transgenic animal. You can see here there's a mouse with a transgenic growth animal having expressing the growth hormone. We can insert foreign DNA in particular species and we can get the results. You can see this fluorescent feces. Okay, they get this fluorescent gene from other species and they can glow in the darkness. So this is an example or one of the applications of recombinant DNA technology. If we talk about its application in medicine, the human gene therapy, we can treat the what we can say inherited disease or the inborn errors by correcting the gene mutation. We will see the complete human gene therapy in our next video, but it's one of the most important technique to treat the inherited diseases. It's helpful for the diagnosis of genetic disorders and certain forensic evidences. So it has a very wide role in the applications in the field of medicine. So you can see this are the recombinant DNA technology, but this is just a joke. But this is the hope with the help of recombinant DNA technology. If we use this technology properly, then we get so many applications that can change the life of human being in case of certain diseases or in terms of treatment or in the diagnosis. So these are the references.